Hello and welcome everyone to this video podcast for evolution and diversity. This will be the first of two different videos that I'm going to present where I'm going to talk about key innovations in animal phyla. In this video, we're going to talk about multicellularity, tissues, symmetry, body cavities, segmentation, and digestive systems. In the next video, I'll talk about a few other adaptations, including muscular, skeletal, circulatory, excretory, and reproductive systems. So let's begin at the top of the list and look at multicellularity. Now, the term multicellularity is not new to you. We've mentioned it a few times throughout the semester. And I'm just going to mention a couple things about it here and how it is relevant as we think about the diversification of animals. So if we look at this phylogenetic tree, we can see that at this node here, that gives rise to all of these different groups of animals, that the trait that they all have in common is multicellularity. All animals are multicellular, and it's important to remember that the ability to be multicellular arose several times throughout evolution. However, when we think about the animals, the evidence suggests that it only arose once, that this one common ancestor led to the diversification of all of these animals listed in the rest of the phylogenetic tree. And as we mentioned in the last video, that does of course mean that animals are monophyletic. So the importance of multicellularity we've mentioned a few times before. And so those same features still apply. But I want to give some examples in the sponges and then jump to humans and talk a little bit about how this multicellularity is really important in these two extreme organisms. All right, so let's begin with the sponge. And as we've said before, the sponge is multicellular. In fact, it has approximately 20 different cell types. One of the advantages of being multicellular is that you can have different cell types that have different specializations. And the sponges are no different. Even though at first glance they don't appear to be all that complicated, they actually have a fairly complex set of cells and processes that they have to do in order to stay alive. So let's look at a diagram of the sponge. So in this diagram, you can see a cross-section of the sponge shown here. We had already mentioned the choanocytes, and you do need to make sure you remember them and what they do. Remember, they're responsible for creating the flow that moves through the sponge in order for the sponge to feed. But you can see these other cell types here as well. The lymphocytes, pinocytes, oocytes, porocytes, amoebocytes, sclerocytes. Now, I'm not going to ask you to memorize all of these different cell types or what they do. I'm mainly showing them to you here to emphasize the point that in order for this sponge to exist in the way it exists, it requires different cell types. It requires cell specialization. But it's only 20 different cell types. So now let's compare this to a more derived animal. And let's look at humans. What I'm going to say for the humans can be said about many of the different animals that came after the sponges. But in humans, we have approximately 37 trillion cells. That's a lot of cells. But we only have 200, approximately, cell types. So we have approximately 10 times more cell types than a sponge. At first thought, it doesn't seem like that's a whole lot more than a sponge. But this has allowed humans and other animals, as they have evolved, to take on more specialization in things like digestion, respiration, circulation, immunity, movement, the skeletal system, the muscular system, and many others that I could list here. Suffice to say that multicellularity has led to a great degree of cell specialization that has been incredibly important as we think about how new adaptations have aided the steps of evolution. All right, let's move to a discussion of this next innovation, or we might say adaptation, that animals have. And that is that most animals have tissues. So let's define what a tissue is. These are groups of cells that have a similar structure and act together to perform a specific function. Within the animals, we distinguish groups based upon the number of embryonic tissues they have. Animals are either diploblast, meaning they have two tissues, or they're triploblasts, meaning they have three tissues. And I should stress, I don't mean 2 tissues or 3 tissues in the adult. This is what forms during the embryonic stage. I should also point out that there are some animals that don't have any tissues. So I'm going to put that up here. No tissues. And this would represent the sponges. They're the only group of animals that don't have tissues. The sponges, or more specifically, porifera. I want to talk a little bit more about diploblasts and triploblasts. And when I'm done with that, I'm going to talk about some of the ways we describe tissues in adults. All right, so let's talk about the diploblasts and triploblasts. And what we're looking at here is a cross-section through that early embryo showing the different tissue layers. And let me identify what the different tissue layers are. The blue here represents the ectoderm. The green represents the endoderm. And the red in the triploblasts, this represents the mesoderm. We'll talk about each of these 3 layers in just a moment. This space here between the ectoderm and the endoderm in the diploblasts, this is a non-living layer. In the trypoblast, that non-living layer is replaced with a living layer called the mesoderm. And then this spot here in the middle that they both have, this will eventually become the digestive cavity. So some phyla are diploblasts and some phyla are triploblasts. And one phylum, the sponges, don't have any tissues. So I'm going to erase the diploblast side here and focus on the triploblast because I want to tell you what the ectoderm becomes and what the endoderm becomes and what the mesoderm becomes. Now, as a frame of reference to understand where this stage of development came, remember that in the animal, you would have had a fertilized egg, the zygote. This one single fertilized egg then will divide multiple times until it is this ball of cells. The cells are fairly indistinguishable at this point. And interestingly, the size of this structure here, which is called the morula, I will never ask you on the exam what a morula is, but just so you know, in case you're ever on Jeopardy and that's a question on Jeopardy. But the morula and the zygote are about the same size. They don't change greatly in size. And that's because the cell divisions occur so fast that they just divide. They don't have a chance to grow. Then this morula stage will form what we call the blastocyst. And the blastocyst, essentially what it will do is it will hollow out the inside of this cell here into this mass of cells in the middle. So here, this is just space. And this mass of cells in the middle, which we call the inner cell mass, I can't imagine asking you that on an exam, but I guess I could. This will become the embryo. The cells on the outside here will go on to become structures like the amnion, the placenta, the yolk sac. So very important structures, but not what will become the embryo. This embryo here in this inner cell mass will eventually go into a stage called the gastrula stage. And that's when we begin to form the 2 or 3 tissues. And though I don't have room on this board, and I'm not going to go through all the stages, but after this gastrula stage, then the embryo will continue to develop, and it will hit other milestones, such as forming certain organs and organ systems, and then forming the fetus, and ultimately the developing fetus will be born. But let's focus on this a little bit more and talk about the 3 different layers. So we have the ectoderm, and the cells in the ectoderm, as they divide and specialize, they will become skin cells and the nervous system. I probably didn't stress this enough, but the reason this is really important is this has already begun to specialize these cells. These cells can become skin cells or nervous system cells, but they can't become the cells that will be the differentiation fate for the mesoderm or the endoderm. They've already begun to specialize. Let's talk about the endoderm next. The endoderm gives rise to the lining of the digestive tract and most of the digestive organs, like the liver. And lastly, we have the mesoderm. And remember, the mesoderm is only seen in the trypoblast. It is not seen in the diploblasts. The mesoderm will produce cells that will become muscles, including the heart and other parts of the circulatory system, bones, and many organs. So these terms, ectoderm, endoderm, and mesoderm, are important to describe this embryonic stage of development. But as the embryo differentiates and the cells specialize, we place these tissues into a different category system. And I want to list those for you right now. And now the new grouping of tissues that we would see in a fully formed organism would be connective tissues, epithelial tissues, muscular tissues, and the nervous tissues. And just a little bit about each of these. The connective tissues, they provide structure and support and are important in cell communication. Epithelial cells cover and line most surfaces within an animal. This would include our skin, digestive tract lining, and the lining of blood vessels. Muscle cells, they have the ability to contract, so they can contract. We think of muscular cells as those associated with the heart, skeletal muscles, and what we call smooth muscles, the muscles that are found, for instance, in our digestive tract. Nervous tissues contain cells that send and receive messages. There's more to a lot of this, and when you take a physiology class or form and function, you'll go into much greater detail of these different kinds of tissues. Next, I want to list the different phyla that either have no tissues, are diploblasts or triploblasts. For no tissues, this is only periphery. The diploblasts include cnidaria and ctenophora. Triploblasts include everything else, and I'll list them here, but it's everything else. Now let's move on to our discussion of symmetry. Essentially, a symmetrical organism is one that can be divided by a plane, so kind of cutting right through it, so that the resulting halves are almost identical. So when we think about symmetry in animals, there's one of three options. The first is there is no symmetry. The second is something we call radial symmetry. And the last one is something we call bilateral symmetry. I want to go to a PowerPoint screen to show you the differences because I think it'll be easier to see looking at some of these figures. So here we see the three types of symmetry. No symmetry, like the porifera, the sponges, and that's not surprising because they seem to be the exception to a lot of these animal adapts. But radial symmetry means there's at least two different planes that the organism can be cut through, and you're going to get nearly identical halves. So if we look at this cnidarian here, we can see that we have one plane. If you slice down through the cnidarian this way, you end up with a half over here and a half over here that are nearly identical. You could also cut the cnidarian this way, right? And now you're going to have a half over here or a half over here, and each half will be nearly identical. You could cut this cnidarian in one of many different ways, and you're going to see that each half is nearly the same on either side. It forms this kind of spoke-like structure. So it has to have at least two planes of symmetry. Okay, now let's talk about bilateral symmetry. Organisms that have bilateral symmetry only have one plane of symmetry. As you look at this arthropod here, it has this one line of symmetry right down the center here. And you can see that from this one plane of symmetry, you have two sides that look nearly identical. There are no other planes along this organism that would produce two sides that are nearly equal. You can't cut it this way, right, next to it, or you can't cut it this way. You don't end up with equal sides. So it's bilateral, only two planes of symmetry. But throughout the course of evolution, animals that first appeared had no symmetry, and then they transitioned to radial symmetry. And it's not until we see the triploblasts, those that have endoderm, ectoderm, and mesoderm, where we see bilateral symmetry. All right, so let me introduce this term to you, cephalization. And essentially this is the formation of the head and the concentrating of sense organs and sense cells in the head region. Having this bilateral symmetry and the formation of all these sense organs in the head allowed for a much more directed movement and a much more efficient hunting. And this allowed these organisms to move into different habitats that previously might not have been very hospitable to them. This allowed them to move into these previously less desirable locations. And this allowed for the spread of animals. As I did with the tissues, I also want to show what kind of symmetry we see in the different phyla. And as I mentioned before, the porifera have no symmetry. And we only see radial symmetry in cnidarians and the ctenophora. All other phyla that we're talking about here have bilateral symmetry. So let's move on to our next adaptation, and that is the body cavity. And we consider the the main body cavity of an animal. We call that the coelom. So let's begin this discussion of the coelom. And as we do, I want to draw a picture of a worm. And I'm picking a worm for a couple of reasons. The first is that it's somewhat simple to draw, and I can do so without too much embarrassment, but also because it's simple enough to highlight some of the features without it being too complex. So as we think about a typical animal that has bilateral symmetry, we can think of it as a tube within a tube. The inner tube is essentially this digestive tract here, where food comes into the mouth and it moves down the digestive tract, and any waste products exit through the anus. And it's important to remember that this is essentially, and supposed to be, a one-way direction, right? So food comes in undigested and moves through the system and gets more digested. And this allows for a nice order of cell specialization to allow for the digestion of complex materials. So this digestive tract, or we might just call it the gut, is shown here, and remember it is derived from the endoderm. Now this outer tube here, the outer layer, this includes the skin as well as the nervous system. The skin is on the outside, the nervous system is right along the inside, which we'll just show here with a slightly different colored marker. And remember, this skin and the nervous system, this comes from the ectoderm. Now, in addition to the gut here and the skin and nervous tissue, we also have the muscles and other organs. And they're going to be distributed in between the gut and the skin. So this will be the muscles, organs, and remember, these are derived from the mesoderm. And because the muscles and organs are somewhat difficult to draw, and they're kind of varied in many ways, I'm just going to draw it as kind of this line through here. Now, besides these three different groups of tissues here, we also have this space here. And it's a space that is lined with mesoderm's derived cells, and it's filled with fluid. This is in essence that space where the organs rest in, and it is this space that we call the coelom. Not all animals have this coelom. Not even all animals with a bilateral symmetry have a coelom. So I want to spend a little bit more time talking about the way we can categorize an organism based upon whether it has a coelom. And then I want to talk a little bit about some advantages that a coelom provides animals that have a coelom. So animals are either called coelomates, acoelomates, or pseudocoelomates. So animals that are classified as coelomates have coeloms that are completely lined with mesoderm-derived tissues. For instance, you and I, we are coelomates. Acoelomates are animals that lack a coelom altogether. They still have the bilateral symmetry, but they don't have a coelom. Anytime you see the letter A in front of a word, it almost always means without. So a coelomate has a complete coelom, an acoelomate has no coelom, and anytime you see the word pseudo in front of a word, it refers to it being a false something. And so in this case, these will be animals that have a false coelom. A more specific way to say this is that these are animals whose coelom is only partially lined with mesoderm-derived cells. So you should be familiar with these three definitions. So what kinds of animals are coelomates, acoelomates, and pseudocoelomates? Coelomates are the following: Annelida, Mollusca, Arthropoda, Echinodermata, and Chordata. The acoelomates only include the Platyhelminthus. Pseudocoelomates are Rotiferas and Nematoda. If you're keeping score at home, you might have noticed that I left off Porifera, Ctenophora, and Cnidarians. And that's because those three groups either have no symmetry, like the Porifera, or they have radial symmetry, like Ctenophora and Cnidarians. Only animals with bilateral symmetry, such as the ones listed here, will be a coelomate, an acoelomate, or a pseudocoelomate. Next, I'd like to list some advantages that are associated with having a coelom, even a partial coelom. The first is that a coelom provides a space for oxygen and nutrients to circulate. Ultimately, this allows for a larger body size. A coelom also enables the internal organs to move independently of each other. A third advantage is that it provides a hydrostatic skeleton. Essentially, a hydrostatic skeleton is a flexible skeleton that is formed by fluid. So it's not like the bony skeleton that we might think of. It is this more flexible one, and it's more flexible, right, because it's formed by fluids. But it does provide protection and flexibility to the organism. Another advantage is that it allows for much greater body flexibility. It also allows for greater size and complexity of organisms. All right, and two more advantages I want to list is that they allow for the storage of eggs and sperm during maturation. All right, and then this last one is that these coeloms help to compartmentalize body parts. Now let's move to this discussion here of the differences between a protostome and a deuterostome. We just got done talking about body cavities, that is the coelom, and how we use that to place different animals into different categories. Another way we can categorize animals is by looking at an embryological development. And what we find is that amongst the animals with bilateral symmetry, that we can place them as either a protostome or a deuterostome. So what's the difference between these two? Let's start with the protostome. Essentially, this is translated to first mouth. And for our purposes, what we mean is that the mouth developed before the anus during embryonic development. Deuterostomes is the reverse of that. That is, deuterostomes translates to second mouth. That is, that the anus developed before the mouth. The vast majority of all animal species are protostomes. Deuterostomes tend to be larger animals, and they also tend to be the predators that we know of. The protostome phyla include the Rotifera, Platyhelminthus, Annelida, Mollusca, Nematoda, and Arthropoda. There are only two phyla that are found classified as deuterostomes. They include the Echinodermata and the Chordata. Okay, let's talk about segmentation next as an adaptation that some animals have. This is essentially the division of body or a part of a body into a series of similar structures. A good example of this would be the segmented backbone of vertebrates. So animals with a vertebrae represent this segmentation event. Another good example, and I'll show a picture of this in a moment, is seen in animals like insects and animals like a lobster that have very specific segmented parts of their body. So what is the main advantage of segmentation? Well, we believe the biggest advantage of segmentation is that it allows for specialization in particular regions of a body. So if you look over here at these drawings of various kinds of animals, you can see how the segmented body has allowed for regional specialization. As I mentioned before, the lobster here, right? It has a segmented part where the head is, where the body is, where the legs are, the tail end here, the region here that contains its lovely claw. Same thing with the scorpion, right? You can see various regions that allow for specialization. Now, we don't see segmentation in a lot of animals, but we do see them in the annelids, the arthropods, and our good friend the chordata. We don't see segmentation in any of these other animal phyla. Okay, let's go ahead and begin this last discussion item for today's video podcast. And we're going to talk a little bit about the digestive system and some of the differences that we see within animals. And there's a lot of ways we could talk about this, but I'm going to center this discussion around incomplete digestive systems and complete digestive systems. An incomplete digestive system is a digestive system with only one opening. In an incomplete digestive system, the single opening serves as both the mouth and the anus. A complete digestive system, on the other hand, consists of a digestive tract with two openings. That is, they have both a mouth and an anus. So what I'd like to do now is show you a diagram of an incomplete digestive system and then one of a complete digestive system. So here is what incomplete digestive systems would look like. You have an animal right here that looks somewhat like a sea anemone, and then this fish is going to be trapped by it, and it's going to be pulled into its digestive tract. And once in there, the enzymes found within the digestive tract will begin to break down this fish into smaller pieces. Some of these pieces will be nutrients, and those nutrients will be absorbed into this sea anemone-like animal, providing it the nutrients it needs to survive. However, not everything it eats is a nutrient. It needs to get rid of that waste, and so it needs to expel that waste outside the same opening that the food came in. Again, an incomplete digestive system only has one opening, food coming in and then waste going out. So here is a drawing of a complete digestive system, one way going from the mouth to the anus. This allows for a systematic breakdown or digestion of the food that we eat. In the mouth, it begins its digestion, and then as we swallow the food, it ends up in our stomach, and through some mechanical and chemical digestion, that food is broken down into smaller pieces. And then as it moves through the small and then the large intestines, more of those nutrients will be broken away from the main particle and be absorbed into our bodies. As shown here, we also have some of these accessory digestive organs like the gallbladder, the liver, the pancreas, that all aid in the digestion of the food as it goes through our system. And then in the end, all of the food that we do not use for nutrients will be concentrated in the large intestines and then in the rectum, and will leave our body through our anus in the form of feces. So there are many advantages to this complete digestive system. And I just want to list a few of those. One is it allows for this continuous processing of new food. Let me back up and explain that a little bit more. So as food comes in, at one time it begins its digestive process, right? And then you could have a meal sitting down here somewhere in your small or large intestines and then continue to feed the system, so to speak, and continue to eat. So there's this continuous process of eating and digestion that can occur. Compartmentalization of different processes. So let's go back to our picture. Each of these areas of digestion, from the mouth to the stomach to the small and large intestines and then to the rectum, all have a different function in digestion. So more nutrients and energy absorbed, and this allows for a larger sized organism. Because of this continuous digestion process and having the compartmentalization of digestion, organisms that have a complete digestive system are able to pull more nutrients out of those food particles. And as I say here, it allows for a larger sized organism. The last thing I want to talk about is the way we can classify different feeders based upon their ecological role. And we've mentioned some of these in passing, but I think it's important to highlight them again. The first are the detritivores. And these are going to include a lot of your worms or millipedes and some other animals, but they essentially will feed on dead and decaying matter. We also have herbivores. And herbivores, these are the plant eaters. Lots of examples of herbivores within animals. Pandas, as you see in this picture, are one example because they eat bamboo. And then we have some animals, and they only feed on animals, like the owl shown here. And then lastly, we have some animals, and humans would fall into this category. Bears would also fall in this category. And they feed on a combination of animals and plants. All right, Again, let's just list the different animals that use incomplete or complete digestive systems. Incomplete digestive systems are the ctenophores, the cnidarians, and the platyhelminths. Complete digestive systems are used by rotifera, annelida, mollusca, nematoda, arthropoda, echinodermata, and chordata. We didn't mention porifera because they don't have either an incomplete or a complete digestive system. Rather, they use a cellular-based digestive system. Again, as we will see as a common theme, porifera will routinely be the outlier, not having all of the same characteristics that most of the other animals have. Okay, that is all I have for today's video podcast. In the next video podcast, we'll continue our discussion of the different animal innovations and adaptations that have allowed them to be so successful. But that is all I have for now. So if you have any questions, please let me know. If not, I will see you in class. Bye for now.